in this video, we're going to talk about DNA repair. There are essentially four mechanisms in which the DNA repairs itself. First is the mismatch repair. Then we have the nucleotide excision repair. Then we have the base excision repair. And then we have the double strand break repair. The first one is the mismatch repair. Now, what does mismatch means? It means that here we have essentially a DNA strand that is replicated, and we have a code for it. So we have A, T, C, G, A, T, C. Now, we know that it's complementary. It's going to be T, A, G, C, T, A, and T. Right? That's the complementary. But what happens in mismatch is that against an A, there might be any other. It might be a C. Or against this C, there might be an A. So that is not complementary to each other, and that is a problem. That is a mistake. In order to correct that mistake, what is called a mechanism, it's called mismatch repair. We have the MUT proteins for this. So these are the MUT proteins that we use, and it occurs within minutes of replication. It's important to notice that it occurs within minutes of the replication. So there are basically three steps, or you could say two steps basically. The first is recognition. You have to recognize that there is where is the a problem. So that, how do we do that? And that's important. We do that by the degree of methylation. So another thing to notice is that when we have an old DNA strand, so, or you could say the parent strand, we have a parent strand right here, and it's old DNA. So old DNA is methylated. The adenines, the A's, have CH3 bound to them, so they are methylated. They are old. But the new strand, because it's new, it does not get methylated that fast. So this is either non-methylated or hemi-methylated, half methylated. So because of this degree of methylation, as you see, there are more CH3 and there is only one CH3 in here. So the MUT proteins will recognize, oh, it has less number of methyl groups. So this is the new strand. There must be a problem here. So that's recognition. And then, so as I told you, the adenines of the GATC sequences are methylated. So that you need to know by DNA adenine methylase enzyme, DAM enzymes, right? Then we have repair. So repair is done by endonucleases that nix the strands. And then we have the exonucleases that removes the nucleotides. So the thing is that here is the backbone, and then attached to it is ATC. So the exonuclease is going to come. The exos are going to nick the strand here. And then the endonucleases are going to come, and they're going to remove it and take it away. Right? That's how it happens. And the gap is filled obviously by DNA polymerase 3. So we have a nick here, and then the endonucleases are going to cut it up. An important consideration is Lynch syndrome. Lynch syndrome is hereditary non-polyposis colorectal cancer, and it is the mutations mismatch repair. Is that they do not have mismatch repair enzymes, or there are mutations, or they're fault or defective, and that is why they cannot repair DNA. So cannot repair DNA, and that is why the individuals are more prone to cancer. So that causes cancer, because we cannot repair DNA. So mutations are going to accumulate, and that is going to cause cancer. Okay, so next is the nucleotide excision repair. The nucleotide excision repair is, uh, the name suggests that there is going to be complete excision of the whole nucleotide, right? So what actually happens is that due to UV radiations, due to ultraviolet radiations, there is pyrimidine dimer formation. Two pyrimidines, like thymine plus cytosine, they're going to form a dimer within the same strand. So right here, if this is T, this is C, they're going to form a dimer amongst itself. And now this bulky dimer is a problem because when DNA polymerase 3 is working its magic, or it's replicating, it is going to get stuck here. The dimer is not going to let it go forward. So DNA replication might be stopped and many other problems. So what do? We first recognize, right? Recognition is first. How do we recognize the dimers? It's by the UV specific endonucleases. We call them UVR-ABC-XE nuclease. That's just a name, right? They are actually endonucleases. And then we have cleavage. So the cleavage occurs at both 3' and 5' strands of the dimer. So at right here and then right here at both sides, it is going to create nicks or cleavage. And then gap filling is by DNA polymerase 3 and ligase. Now, another thing to know is that, okay, I may have skipped this one, sorry. Transcription coupled repair. Transcription coupled repair means that it occurs at the level of mRNA in eukaryotes, not in prokaryotes. Okay, the same mechanism, but it occurs at the level of mRNA, not DNA. Now, the problem or the clinical vignette is xeroderma pigmentosum. Now, what is xeroderma pigmentosum? It is defective NER. You have defective enzymes for nucleotide excision repair, and that is going to cause your DNA to be more prone to UV lights. It is going to be more prone to mutation by ultraviolet radiation, and that is going to cause cancer. So it's important. It's a defect in NER that leads to DNA damage by UV, and that causes cancers, different skin cancers, etc., etc. Okay. The next is the base excision repair. Now, the name suggests the base is problematic, not the whole DNA molecule. Okay, the base is problematic, and we remove the base, right? So for removal of the base, now how are some ways that a base can be altered? It could be either because of spontaneous deamination or usage of deaminating or alkylating agents. For example, nitrous acid, they convert cytosine adenine into hypoxanthine and guanine into xanthine. And there may be alkylation of adenine by dimethyl sulfate. So these are some examples that you might need to remember. Now, what do we do? How do we solve this problem? The first step is base removal. We remove the base by DNA glycosylases. Now, this will leave an AP site. Now, what is an AP site? An AP site, as seen right here, usually this is the molecule right here in front of you, and this is the pyrimidine, or it could be a purine. So by glycosylation or different by the action of glycosylases, we remove this. Because we remove this space is now empty. This empty space is called as an AP site, antipyrimidinic site or antipurinic site. Then we recognize the AP site. The recognition of AP site occurs by AP endonucleases, and then they are excised. The whole molecule is then excised. Why do we excise the whole molecule, because gap filling is done by DNA pol 3 and ligase. There is no other way to magically attach a base to the molecule. We need to excise the whole molecule. The whole molecule will be sacrificed just because the base is defective. That is base excision repair. The next and the last is double strand break repair. It basically means that this is the DNA strand right here, and there is a break, and we have to join the both strands. So this is double strand that is breaking. So there are two things. We do not need to know behemoths of information on this. We just need brief stuff. One is the non-homologous end joining, NHEJ, which occurs. This is a procedure, non-homologous end joining. But in this case, DNA might get lost, and it is very much error prone and mutagenic. Then we have the homologous. The other thing is the homologous recombination. This is the same as the genetic recombination that occurs in meiosis, right? So this is the same mechanism, and it occurs in late S phase or late G2 phase, while NHEJ can occur anytime. Okay, and HR is more error-free. Now, an important clinical vignette is that mutations in the BRCA1 and BRCA2 genes are basically mutations in the HR gene, homologous recombination genes, that can cause breast and ovarian cancers. And that is it for DNA repair. Thank you so much.